Well, first of all, thanks for having me here, and I'm glad to share that I can take this off because I'm negative as of 5 minutes ago, which also means I can't check in for my flight back tomorrow. Yep. Let me see. Should be on. Resume slideshow. I was trying. Okay. It's working. Yep. Okay. Disclosure first, funding and some disclosure that will come in handy by the end of the presentation. And additional disclosure, when I say we during this presentation, I will say that a lot. It's actually these people who are in the lab. We got the work done. Mostly professional writer these days, either grants or papers. And in addition, I have to thank a lot of collaborators and all around the world, in particular here, a few colleagues for the presentation that is relevant today, both in the United States and Europe. A few years ago, I was honored again to be invited here, and I tried to convince you that a lot of things change in packed red blood cells as a function of storage duration, and that these things that change can be cataloged through omics approaches, mass spectrometry-based proteomics and metabolomics, for the most part in my lab. And all of this is ultimately summarized in what I think is a rather comprehensive review I had the pleasure to work on with Dr. Tatsuro Yoshida and Michel Podin. And just to simplify the main take-home message from our omics studies, our blood cell storage, we come up with a relatively rather simple model that posits that during storage in the blood bank under a refrigerated condition, the cold temperature have a negative impact on the activity of proton pumps and enzymes because of thermodynamics. And glycolysis slows down. As that happens, high-energy phosphate compounds, ATP and DPG, which are relevant to regulating red cell hemoglobin oxygen offloading. As you consume these small molecule metabolites, as you're not making much more of those, you actually increase hemoglobin oxygen saturation, and through Fenton-Abreu-Weiss chemistry, you start accumulating reactive oxygen species. Ultimately, you start accumulating this oxygen and stress to proteins, small molecule metabolites, and lipids. And what I also showed you last time is that this oxygen and stress is actually a sort of relay switch in a key red blood cell metabolic enzymes, glyceraldehyde 3-phosphate dehydrogenase, and that oxidation of GAPDH and/or migration and binding, inhibitory binding to the N-terminal cytosolic domain of BEN3 is a sort of relay switch to block late glycolysis and redirect glucose oxidation fluxes towards the pentose phosphate pathway to generate reducing equivalents, such as NADPH, which is the most important reducing cofactor in red cells to regulate glutathione homeostasis and glutathione-dependent antioxidant enzymes. Alas, the oxygen stress and the capacity to make NADPH are not balanced, and oxygen stress is usually overcoming these antioxidant systems. And ultimately, red cells start accumulating irreversibly oxidized proteins and lipids in the membrane and start vesiculating them, progressively losing what is their viscositic phenotype and progressively accumulating an echinocytic and spherocanocytic phenotype, ultimately a spherocyte phenotype, which prompts them for splenic sequestration. In older papers of ours and other groups, it has been shown that by the end of the storage period, by storage day 42 in most countries around the world, but at least in the United States, you have, and in Italy, these are my two experiences, I would say most Europe except for Northern New Europe, by the end of the storage period, you have approximately 15 to 35% of red cells have accumulated a irreversibly modified spherocanocytic phenotype with irreversibly altered morphology. It happens that more recently, elegant work by Camille Roussel in Pascal Amiro's lab in Paris, they actually showed that these red cells with the increased morphological alterations, especially reversible alterations, spherocanocytic and spherocytes, are preferentially sequestered in the spleens in vivo upon transfusion. And they called this smaller red cells, smaller because of the vesiculation events, small microerythrocytes. What we did now in collaboration with Pascal's group is that we can actually use fluorescent-based approaches to label and sort The SMEs, the erythrocyte at the end of storage, have extreme morphological alterations from red cells that at the end of storage has still a preserved morphology, and compare those to fresh red cells. And if I try to convince you in the past that storage is characterized by the accumulation of a series of lesions, in the next few slides I'll try to convince you that the opposite is also true, that if you have, like we do have now, a strategy to sort end-of-storage red cells that are morphologically altered, we now see that all the things that can go bad during storage do actually go bad in the population that looks morphologically altered irreversibly, but not in the end-of-storage red cell population that is morphologically still preserved. Indeed, I don't think you can see the pointer here, but the red cells that in end-of-storage are still preserved morphology cluster right on top of the fresh red cells, while the others are clustered separately. And I will spare you the details of this, but energy metabolism is reversibly compromised in the morphologically altered SMEs, but not in the long-storage red cells with good morphology. The same goes for the capacity to activate the pentosphosphate pathway, lost in the morphologically altered red cells, but not in the morphologically preserved red cells, and the same goes for glutathione homeostasis. Hemoglobin oxidation, irreversible oxidation of histidine and 93 and cysteine 94, if you count anhydromethionine, which are relevant to hemoglobin function in carrying and delivering oxygen, but also of a series of other enzymes, including, for example, glyceraldehyde 3-phosphate dehydrogenase, and a series of critical antioxidant enzymes. The same holds true for lipid peroxidation, phospholipid exposure, and lysophospholipids, whose accumulation is higher, significantly higher in the SMEs, morphologically altered cells, but not in the other groups. And the capacity to repair oxidally damaged lipids through the Lens cycle, which leverages acyl-carnitine, acyl-CoA pools to restore the oxidized and phospholipase 2 fatty acyl chopped lipids into lysophospholipids back to the phospholipids. This acyl-carnitine pool that participates in the lipid damage repair mechanism through the Lens cycle is completely drained in the SMEs, but not in the other groups. All this information is to say that now with this omics data that we have about red cell storage, we can, and we did this in collaboration with Sebastian Himbert and Dr. Einstatter here in Canada, we can generate this in silico computational modeling to, based on omics data, predict the deformability of red cell membrane, which we actually test at the end of storage, and is indeed decreased by the end of storage and could contribute to explaining increased splenic sequestration upon transfusion in vivo. We can also go a little bit of a step further. One thing that I usually hear when presenting this data is that, well, okay, all these things happen in storage, but upon transfusion in vivo, some of this storage lesion is reversed. What we can do when we did this, actually this paper got accepted in a kind of a weird way in Blood Advances 2 hours ago, in collaboration with Dr. Robin van Bruggen. We can biotinylate red cells with different biotin contents, low biotin versus high biotin, for fresh red cells and end-of-storage red cells, transfuse to the same healthy autologous recipient, and then recover red cells at 1 hour, 6 hours, and 24 hours from transfusion, and determine to what extent the metabolic storage lesion, at least, is reversed upon transfusion in vivo. Of course, it is to be acknowledged that there is a survival bias in that the red cells that are not sequestered in the spleen are the ones that supposedly are, if I convince you with a SME story, the ones that are less damaged in the first place. But as you have the capacity to recover these biotinylate red cells in vivo and look progressively at the changes in, for example, ATP, DPG, and pentosphosphate pathway marker ribose phosphate, you see that they are indeed recovered over 24 hours in vivo, but at a rate that may not be sufficient to, for example, make the red cell resistant to hemolysis, intra or extravascular hemolysis in vivo in a non-healthy, non-autologous recipient, where you also have all sorts of inflammatory and oxidative stress-related complications ongoing. And also, even 24 hours are not sufficient for some pathways, such as the pentosphosphate pathway, the one that I told you is relevant to counteract oxidant stress in the first place during storage. it is not fully recovered even after 24 hours from transfusion. Pentose pathway that actually does correlate is actually the top correlate along with the high-energy phosphate compounds like ATP and DPG. Among the top metabolic correlates to post-transfusion recovery. So if you were to measure what percentage of red cells still circulates at 24 hours from transfusion, the metabolic markers of those red cells that could predict post-transfusion recoveries are indeed metabolites in this pathway. We can also use this metabolomics analysis to go a little bit further from a functional standpoint in that the fact that red cells circulate does not necessarily mean they are functioning. So we can actually measure oxygen kinetics, and we did this with Pavel Sviatak in Oxford, with a single-cell oxygen kinetics macrofluidic chamber, where we can test the time it takes for red cells to uptake red cell hemoglobin to upload and offload oxygen at a single-cell level. And what we do find is that storage actually compromises, to some extent, this capacity by the alpha second to 0.7 second scale in a second scale. So a significant contribution in slowing down oxygen floating capacity to red cells. And of course, a lot of this has to do with ATP and DPG, which are historically known as metabolic mediators of red cell oxygen offloading capacity. But we have more advanced metabolomics approaches. We can find many more correlates that are predictors and contributors to, in interventional studies I don't have time to show today, to red cell capacity to offload oxygen. So if all of this is actually true, a lot of things change with storage, and these things are relevant to red cell post-transfusion recovery and performances, such as oxygen kinetics. Why is that the clinical trials are not showing significant differences, at least when comparing standard of practice versus selective transfusion of the freshest available units? One tentative answer to that is that blood might not necessarily store the same across all people. Some donors, healthy autologous donor volunteers, actually store consistently very well, and their post-transfusion recovery is consistently above the 75% threshold for European Council and Food and Drug Administration guidelines for storage quality. But some donors actually consistently store really poorly. And animal models, in particular, again, work by Dr. Zimring's lab, showed that even genetic heterogeneity across different murine models of red blood cell storage and post-transfusion recoveries have different post-transfusion performances. So genetic underpinnings may contribute to explaining this phenomenon. I probably showed this a few years ago, and I will show you where we went forward from this. But the take-home message here is that, yes, red cell age in the unit, and things age and go bad as blood storage progresses, but blood-like people might not always age the same, and some people age really well, some people not necessarily so well. And the age of days, the age in the chronological age of the units, like days elapsed since the time of donation, may not necessarily mean much from a biological qualitative standpoint. This may not be a political statement. They don't let me back in there. Well, I guess they do now. That didn't go down well in Mississippi, by the way. So how do we go about investigating the impact of genetics and donor biology in general with respect to the storage lesion and post-transfusion performances? I had the pleasure to be involved in the study led by Dr. Mike Bush, the recipient epidemiology and donor assessment study, where the main goal was to investigate factors contributing to heterogeneity in hemolytic propensity of stored red cells across large blood donor populations. Specifically, they did enroll approximately 13,400 healthy blood donor volunteers across 4 different blood centers in the United States, and they tested them for the propensity of red cells to hemolyze spontaneously or following oxygen to osmotic insult, and then they fully genotyped these donors. These are just an overview of the hemolytic tests that were used at this time. Interestingly, these blood donors were then ranked in the 5th and 95th percentile according to their distribution in hemolytic propensity for each one of these parameters. And the 5th and 95th percentile lowest and highest hemolyzed were asked to donate a second unit of blood. And this unit was stored again. And at the end of storage, these parameters, hemolytic parameters, were tested again. And we do see that for some of these parameters, there is a strong reproducibility across multiple donation. So there may be some specific donor-specific factors that contribute, for example, to oxidative and specifically osmotic hemolysis. In the meantime, we had developed high-throughput omics approaches that I didn't have a chance to show you the last time, which make it so much easier for us to analyze samples not in the 5 or 10s, but in the hundreds and several thousands. I will show you, this is just a preview of the Rats3 study where we process 600 samples. And you see in this map from the top to bottom on the left side, the storage duration, storage day 10, 23, and 42. And you still see metabolites that accumulate with storage, so they increase and accumulate in the unit as a function of storage duration. And the right-hand side of the heat map, you have metabolites that are consumed from the unit. They're metabolized as storage progresses. But now you also have a series of metabolites in the middle that are relevant to other parameters that are not just storage dependent, storage duration dependent. For example, we start seeing the impact of sex dimorphism as a function of storage duration, of donor age, donor ethnicity, whether the donor is even on testosterone replacement therapy that requires blood donation as an intervention, blood donor BMI. All these factors do impact red cell metabolism and hemolytic propensity. If you're interested in more details about these studies on top of the related publications, we are now generated an online platform portal. It's called Mirage, Metabolic Investigation of Red Cells as a Function of Aging, Genetics, Environment, and Storage. This platform is going to be user-friendly. Just go online and you click, for example, on the effect of additives. And you will, in this case, we had additive solution 1 versus 3. And you will see metabolite that change specifically as a function of additive, not just additive solution components, such as, for example, as a positive control, citrate, which is more abundant in mannitol-free additive solution 3, but also a series of other metabolites. You can click on the effect of age and identify metabolic correlates to blood donor age, and other factors such as, for example, donor sex, and identify, for example, biology that has been known for decades, such as, for example, female red blood cells have higher levels of creatinine and creatine because they tend to be overall younger because creatine transporters are progressively degraded in red cell membranes as a function of red cell age. It happens that in the meantime, we had the opportunity to collaborate with Jason Acker here in Canada and separate red cells in a unit, in fresh red cells and stored red cells, according to their population and on the basis of density gradients, and identify that overall the female red cells have higher levels of creatinine, especially in the younger red blood cell population that was more abundant than in male. And these red cells tended to be more resistant to osmotic stress, oxidative stress, spontaneous hemolysis, and had a lower degree of morphological alteration and better preserved oxygen managing capacity by hemoglobin. And of course, they had higher levels of antioxidants, including the repeated multiple times, reduced glutathione and pentosphosphate pathway. What we can do with this now big data from multiple omics approaches that we can use metabolites as quantitative traits, not only to determine what are the factors, genetic factors contributing to hemolytic heterogeneity, but also genetic factors contributing to the metabolic heterogeneity of red cells. And since we now have metabolic markers of post-transfusion recovery, metabolic markers of oxygen kinetics, we can, for example, determine the genetic underpinnings, the polymorphic, what polymorphisms in the genetics of the blood donor population impacts metabolites that are relevant to functional aspects of red blood cell storage and post-transfusion performances. We can also, of course, focus specifically on hemolytic propensity. In this very case, Dr. Greer-Page focused on glucose 6-phosphate dehydrogenase polymorphisms, as they ended up resulting from this Manhattan plot, the top genetic predictor of oxidative hemolysis, meaning the propensity of red cell stimuli following oxidant insults. If I convinced you in the first part of the presentation that this is the mechanism, the activation of the pentosphosphate pathway is the mechanism through which red cells cope with oxygen stress during storage duration, G6PD, glucose 6-phosphate dehydrogenase, is the rate-limiting enzyme of this pentosphosphate pathway. And in G6PD deficient people, which happens to be around 500 to 600 million people around the world, you don't have activity, you have significant reduction in the activity of this enzyme. This means that not only these subjects cannot activate this pathway during storage, but they actually have lower levels of this enzyme activity in the first place at the time of donation, which results in metabolic phenotype, especially the pentosusate pathway redox level, that are completely compromised even early on during storage, and they are irreversibly compromised by the end of the storage period at a much faster rate than in healthy blood donors. Ultimately, this translates, and we did this in collaboration with Richard Francis, Saldad Hodd, and Sis Pitalnik at Columbia. We show that especially African-American variant of G6PD deficiency is associated with a poorer post-transfusion recovery of restored red cells from these donors. If you're interested in that, we actually now generated with Jim Zimring a mouse model of med- variant, so less than 1% residual activity of G6PD. That recapitulates phenocopies, what happens in human with the Mediterranean variant, and we are about to publish now a follow-up A-, conditional med-, and humanized G6PD mouse models. And this is what we can do now with high-throughput approaches. This is actually a slide that wasn't part of the presentation, but I couldn't upload my slides, so I had this yesterday afternoon. We now have a chance to put our hands on all the Rats3 blood donor population. These are just the recalled donors, the 5th and 95th percentile blood donors, depending on hemolytic propensity, oxidative, osmotic, and spontaneous. And now you can start seeing that the things that change as a function of storage duration are not as clear as they are when you study, for example, 5 units of red cells. Rather, donor biology is impacting these storage metabolic phenotypes much more than we would have anticipated from very well-curated small cohort studies. For example, we can start seeing beyond genetic factors of donor biology like sex, G6PD status, and so on and so forth, other factors that are relevant to the metabolic lesion that occurs and accumulates during storage in the blood bank. Of course, even in 2,000 samples, you have the metabolic markers of the storage lesion, things that decrease and things that increase, still decrease and increase, but in some subjects, the day 42 levels of lactate are the day 20 level or 10 level of lactate in another subjects. Of course, we can still distinguish additive solutions, even though we were blinded in the study. It's hard to lie to a mass spec. But we can also start looking for small molecule metabolites that are impacting G6PD activity, for example, pentosusate pathway activation, such as the donor smoking habits, exposure to alcohol, drinking coffee. I may have not come up with this idea when being in the countryside in Italy, surrounded by smoking mother, drinking cousins, and lots of coffee. In addition to that, we actually start looking for small molecule drugs, over-the-counter drugs, or prescription drugs that are not ground for blood donor deferral, at least in the United States. And we can see that they are detectable in blood units, and they actually, from blood pressure medication to, acetaminophen, ibuprofen, very common over-the-counter drugs, statins, diabetes drugs like metformin, all the way to Viagra in 3 blood donors over the age of 75. How do we know that those metabolites are actually the drugs we think they are? Because we can actually incubate red cells with an FDA standard library of all the FDA-approved drugs. I'm just showing here for simplicity 1,366 drugs. And not only validate that these drugs are potentially uptaken by red cells, but also are metabolically active in red cells, to the point that we can now build with Synovia Biosciences, here represented by Dr. Arash Porbar and Bernard Paulsen. We can build a map of red cell metabolism as a function of how it is impacted by specific drugs. This could be relevant, for example, when trying to come up with novel additive solution for blood storage, for improving the quality of stored red cells, because now we know markers, we know which pathways need to be activated or downregulated to boost post-transfusion performances or oxygen kinetics. And we know also that drugs are present in red cell from healthy blood donors or not so healthy blood donors, and from these in vitro studies, and we can use all of this to pretty much come up with different compositions that minimize, for example, lipid peroxidation and boost energy metabolism and antioxidant capacity. With this, we actually develop a high-throughput platform that is up and running, and we are testing, like in parallel, I think a few thousand additive solutions per week, for which I disclosed that we received some funding from NIH and HLBI. And with that, I will be happy to take any question. Sorry if I went too late.
So we're open for questions.
Hello. Is there an age limit for donating blood in North America?
Not as far as I know. I had 2 blood donors age 93 in that cohort, so which is pretty impressive. I know that in Italy there are some limitations. For example, my father has been a routine blood donor all his life, but he just been discouraged from donation like last year, age 65.
Yeah, in Canada, we used to have a hard stop at age 71, and we've gotten rid of that. And as long as your personal physician says you're okay to donate blood, we'll take you.
And one other question, you were studying different drugs. Is there any study with testing with marijuana?
As a matter of fact, marijuana is legal in in at least 7 or 8 states now in the United States. The first one of the early adopters was Colorado, where I'm from. And we do see units, there are, CBD or THC or combinations thereof positive. And it does have an impact on some of this pathway.
I think Jim Zimmering saw that in his platelet study too, right? There were donors who've been smoking dope. Dr. Prasdale.
Are these donors screened for alloantibodies and possible effects?
This is above my pay grade, so.
No, I wouldn't have thought so. So Angelo, I have a question for you. Do you, in all of this data that we're starting to accumulate, do you think we're going to get smarter about how we screen donors? Yeah.
My dream would be to have like a personalized transfusion medicine or personalized metabolomics approach. We already have transfusion medicine has been personalized since day one, right? Yeah. At the very least with ABO group, antigen match, and so on and so forth. But yes, ideally from the same blood drop, from the same sample that goes for genetic testing once, that's what Mike Bush wants to do, right? Yeah. The precision transfusion medicine array for genetic characterization each one of the donors. And then you do once, and technically should be it. But from a metabolic standpoint, we can actually get from a blood drop at the time of donation. And the central lab, for example, for Vitalant is in Scottsdale, Arizona. When they test for nucleic acid testing, they could have a mass spec next to it. Now we actually, depending on sample volumes, if you talk about 1 million samples a year, we can reduce the cost of the analysis down to $1 per unit, which makes it feasible on a yearly basis. And pharmaceutical companies that actually want to have access to the data would actually be really, really interested in subsidizing the effort. So I think it's going to happen. There's the interest by Vitalant. That's why we're opening a lab there.
Yeah, no, that makes a lot of sense.
Yeah, thanks for that wonderful presentation, Angelo. I'm wondering if you could give an update to that question from the RED study. Like, have there been donors that's been identified that can really have really great outcomes for recipients?
Yeah, and again, I'm not taking credit for something that I've been, had just invited to join when the Rats project Rats3 at least was finishing, and I was part of Rats4 just for the central lab part. But the answer is, to some extent, yes. Especially, there's some beautiful work from Narek Rubinian based on hemoglobin increment in recipients of these units, because one thing that it specifies that for those 13,400 donors, the satellite unit that has the same composition and surface to volume ratio for red cells and additives was used for genetic testing and metabolomics, but the actual unit went into recipients, non-healthy, non-autologous recipients. And we have the recipient database. So now they're changing name. I think now they're going to call it V2V or vein-to-vein database. The previous iteration of Rats3 was called the Kaiser Permanente Donor Recipient Linkage Database, a mouthful. But yes, they have, based on this information, a few ideas on specific either genetic characteristics or personally I'm interested in the metabolic characteristics that could be predictors of super donors. And again, I probably oversimplified your question because super donor may also mean like donors that donate multiple times without becoming anemic, right? So there's a whole like branch of study there that now I think Eldad Hod is leading with Tamir Kanias and Eric Gurubinian that focuses on the impact of repeated blood donations and with and without iron supplementation. It goes without, I didn't mention that, but ferritin levels were measured for these donors as well. So we have, and I'm applying for funds to run ICP-MS for trace element analysis, so iron levels, but also I'm interested in, for example, lead, cadmium, and so on and so forth. I have the instrument there. It's just a matter of funding to make that happen.
Thank you.
So thanks for a great talk. Can you hear me?
Yep,
Yeah,
thank you.
So I'm wondering if you're planning on doing any kind of interventional studies. So in the sense that like, you know, you're seeing glucose go down or you're seeing lactate go up and so on and so forth, and to try to like treat these red blood cells in a particular way, maybe based on a combination of pathways to try to preserve them longer.
Yeah, not only that, but we actually built like in silico models that we can now use based on this information to predict how metabolism is going to respond. But now, since high-throughput metabolomics actually become cheaper than hiring bioinformaticians, we are also using the brute force approach, where we now screen like over 1,000 of these interventions a week, and then the top candidates actually move to the next step, and then we do combinations, and then we do obviously across different concentration ranges to test those response, and then from the combination, the screening, those response and combo, we can tell which ones move on to the next step. As part of this grant, the SBIR, Small Business Initiated Research or something along those lines, that we got with the Synovia Biosciences one and one with the Hemanex Inc., the anaerobic storage guys. We're actually developing a solution for standard red cells, normoxic red cells, but also for oxygen-depleted red cells, because we know that removal of oxygen from the unit requires a cell metabolism towards specific pathways. So we're trying to feed specifically those pathways with added solution tailored to maximize the quality of hypoxically stored blood. So the short answer is yes, we've been doing a lot of that. And as a follow-up of that, of course, we're going to have animal models. At first, with just post-transfusion recovery, then trauma hemorrhagic shock, and resuscitation. Not very dissimilar from the porcine, the swine model presented by Dr. Cow, which is actually the follow-up part of the study in collaboration with Pedro Cabrales in UCSD. And the final studies that are planned as part of this SBIR are going to be the FDA-required post-transfusion recovery studies with Jose Cancelas in Cincinnati.
Fantastic, thank you.
Great. Thank you very much, Angelo. That was really interesting.